Thank you very much for the invitation and also the introduction. I think today we have witnessed a lot of nice work on cancer research and also extremely powerful technology from 3D image and all the way to single cell technology and also the AI. And I'm actually a chemist, analytical chemist working on methodology for proteomics. And I think this is a powerful tool complementary to genomics. So today I want to share with you how proteomics can help to understand the oncogenesis mechanism for cancer patient and how that can be implemented for the future technology for early detection and early treatment. I think today we have a lot of questions. Why some patients have a poor outcome and some patients don't? So there are actually a lot of genetic factors that may contribute to the different mechanism of the cancer initiation and progression. For example, we may have the different genetic background, and we know that Eastern and Western population have very different phenotype. And we also have the different lifestyle, like the smoking and or the food. And on the other side, we also have the very different environment. And unfortunately, for example, air pollution is a very important factor for East Asia. So from all these factors, they may synergetically contribute to the initiation of the cancer. But on the other side, also think about this one. We all live in the same environment, right? But some people develop into cancer, but some don't. So that suggests air pollution or the other factor may not be the only factors. So everybody may have the different endogenous factor altogether with the environmental factor to cause the cancer. So how can we utilize the new technology to understand what is happening at the patient level? That could be very important to further inspire new strategy development for clinical settings. And I'm working on proteomics. And this is the new field about 15 years later compared to the human genome. And so far, we already decode nearly more than 18,000 proteins. But still, there's a lot of missing protein. About 7% of the protein is still missing. And that includes the GPCR that Dr. Tong mentioned today. But even that, this is a very rich resource that we are able to study what is happening for the disease, for the life. And in addition to the protein expression, protein modification is also very critical. And we already know that there are more than 200 different kinds of protein modification. So altogether, this contribute the complexity of the life, including the human disease. So for example, if I use the lung cancer as the example, we know that EGFR is the important driver mutation, right? And proteomics is able to tell you what is happening of the different subpathway, and that also include the protein modification, especially protein phosphorylation, that is critical for signal transduction. And outside the cell membrane, there's a heavy layer of the glycosylation that also contribute to the important cell-cell interaction, including the tumor microenvironment. So, so by this kind of technology, we actually can provide a very systematic view about what is happening for every patient or the cell. So this is the result. We try to compare the primary cell line, a primary cell from the patient with sensitivity or resistance to the tyrosine kinase inhibitor. So you could see that on the outside, like EGFR, there's a lot of modification on glycosylation, and that also impact the intracellular signaling from phosphorylation. So this could be a very useful resource for to inspire further study for the cell research. And a few years ago, I was very fortunate to have the opportunity to launch the Taiwan Cancer Moonshot. This is a project we work together with the United States. And last year, we renewed the MOU. So initially, the project is only about the proteogenomics analysis for our patient. And when we renew this MOU, we further expand the proteogenomics into more experts, including the policy, including the research, and the other clinical applications. So we are very expecting that continuation of the Taiwan-United States collaboration can align with the goal from President Lai, Health Taiwan. So we hope that we can contribute also to his goal. I'm going to share with you the very first study we published a few years ago. And I want to share with you how do we continue after the academia publication. This is the first project that we try to understand what is happening for our patients who are mostly never smoker. And in Western populations, smoking is the most important etiology, but our patients do not smoke, especially for female who is nearly 100% never smoker. And I don't have time to go through, but basically we perform very deep proteogenomic profile. So we confirmed that we had very different genomic background compared to the TCGA patient who are mostly smoker, male from the Western population. And we also identified the endogenous and environmental carcinogen for our patient. And today, what I want to focus is we discovered that proteomics is a very powerful tool that correlates with the patient's clinical outcome. And from that, we further identify the high-risk patient who develop early recurrence. And in this study, I think one of the most interesting discovery is what is the etiology to cause the initiation of the lung cancer in Taiwan. So our team tried to perform the somatic mutation profile and compare the Taiwan cohort with the TCGA. And among the comparison, we identified three common etiology, including the pH. Please keep it in mind because later we have a very interesting analysis in the later of my talk. But in addition to the three common somatic mutational signature, we also identified two additional mutational signature, including the nitro pH, which is closely associated with the air pollutant like PM2.5. And another one is nitrosamine, which is enriched in female. So this is the very first time that we are able to confirm this kind of environmental carcinogen do have the imprint in our patient at the tumors. And the second question is how, in addition to the etiology of the genomic profile, which may be helpful to identify the carcinogen for prevention. So far we have a project working to the local database for the environmental carcinogen. But the second question we would like to ask is, we really hope that we have a better method to identify the high-risk patient who develop early recurrence. So let me share with this is a question raised by Professor Yang many years ago. I have the collaboration with him for more than 10 years. And every time he told me, oh, Yuju, the EGFR has two driver mutation, but the two driver mutation have very different outcome. The LA5R point mutation have the tendency to develop brain metastasis. And the patient also have the earlier development for resistance to the EGFR target therapy. So overall, the patient with the LA5R has a poor outcome compared to deletion 19. But because they don't know the reason, so treatment strategy is very similar for the patient of these two types. So we try to see if protein is executing function in the cell, that might have better or closer correlation with the patient's outcome. So we analyze the patient's proteome, and we use the proteomic profile to categorize the patient into three groups. And when we compare the classification with the clinical features, it's very interesting to see that all of the aggressive phenotype, including the pleural invasion, lymph node metastasis, and the patient at the later stage, all gather in the green subtype. And on the other side, the red and the blue subtype have a very early stage. So we thought about the idea whether we can combine this kind of molecular classification with the clinical staging. So for example, if the patient is if the patient is in the green subtype, that means the patient tend to have the similar profile with the patient with the late stage. So we call this kind of subtype, we call late-like. And so based on this, you could see that for the stage 1B, we see a lot of patients from the ALA5R are in this subtype. So we thought that maybe but this is not evident in the stage 1A. So we thought that maybe the EGFR start to be different for these two types of mutation at the stage 1B. So Professor Zhang Chen performed another independent cohort analysis, and you could see that LA5 patients do have the poor survival compared to the deletion 19, but this is not evident in the stage 1A. So this suggests that it's likely proteomics can differentiate the different outcome of the EGFR mutations at the stage 1B. But this is a prospective study. So after the paper was published in Cell, I was so nervous because I don't know whether this kind of prediction is true or not. Maybe it's not true. And whether this late-line subtype really develop early recurrence. And 4 years later, in 2023, we received the follow-up data for the same patients. And it's kind of amazing that you could see that in the stage 1B, the recurrence rate is 43%, which is so close to the patient in the late stage. And keep in mind that the patient in the late stage received treatment. So this suggests that the 1B late-line patient is really aggressive compared to the molecular defined good stage 1B. And stage 1A is even more profound, which is about 5-fold higher recurrence rate compared to the good stage 1A. So this kind of confirms that proteomics approach may be a good classification to really identify the high-risk patient. And I skipped the detail. We have a lot of work to identify the potential biomarker from tissue all the way to serum. So far, Professor Yu already have a full protein panel, and he further developed into the ELISA kit. And we are expecting to do this testing on another independent prospective patient cohort for 250 patients. And I'm also very happy that because of the finding, so Dr. Chen Jinxing has a consensus meeting. So they, based on this result, they feel that we should have the urgent need for biomarker at stage 1A and 1B. So this is our goal, and hopefully we are able to push to identify all these high-risk patients. They should receive early treatment and early management or the follow-up. And last year, AstraZeneca also launched their new study result. So from this clinical trial, they confirmed that the target therapy could be further pushed to stage 1B. So this result is kind of aligned with our finding published in 2020. And I actually asked AstraZeneca whether they want to perform the study for stage 1A. And they say yes, but the result will be coming out 8 years later. So I think this kind of companion biomarker could be useful to identify the high-risk patient instead of the general screen treatment for all the stage 1B and the 1A. And after this, we start to wonder whether the late-line subtype is only present in our cohort or it also present in other independent cohort. And when we published the paper, this is actually a side-by-side publication together with the United States. And this is a very nice experience that we see different patients. So we can compare our patient with their patient. And so after this work, the United States feel that Taiwan may be a good team. So they wait for our second study to finish in 2021. And since then, we start to work together. So this is likely the largest cohort for this kind of very deep proteogenomic analysis. So altogether, we have about 400 patients. With this global cohort, you can ask a lot of questions the effect of gender, the effect of region, environmental carcinogen, or the effect of race. So I'm going to share with you about the very fresh data. So we also see this kind of environmental carcinogen in the global cohort. And as you can see from here, the different color means the different carcinogen. The top three, including the nitrosamine, mostly occurs on the never smoker. And the nitro pH and the pH occurs on both smoker and the never smoker. So from this, you could understand that it's actually a very complex process. It's not just the environmental carcinogen. It's not just the genetic variation. It's a combination or a synergetic effect from both endogenous and environmental carcinogen together for every patient. And they have a very different contribution at the individual patient level. And you could also see that for the high nitrosamine, it's correlated with the poor survival. But among all the environmental carcinogen, the ApoB mutational signature and the pH, nitro pH, has a poor survival. So the next question is, we want to ask why these different carcinogen may promote different degree of the carcinogenesis or at a different patient. And we know that this kind of environmental carcinogen start from the long-term exposure at the normal tissue, right? So we do see, I don't want to go through the detail, but we do see that the different carcinogen may go through the different metabolism enzyme, and they induce different kind of degree of the inflammation. And among the three carcinogens, we find out that the nitrosamine is mostly enriched in the female. You could see that most of them are female and smoker, and they have the EGFR mutation. And for the nitro pH and the pH, these two, sorry, I forgot to mention, nitrosamine is mostly used in the food preservation industry. And pH is well known as a smoking signature. And nitro pH is likely from the air pollutant, but according to the literature, it has more than 1,000-fold of higher potential to cause cancer compared to the pH. And very interesting that you can see that pH mostly enriched in the smoker and also from the Western population. But for the nitro pH, it mostly enriched in the male and never smoker. So that seems to suggest that our patients who are mostly non-smoker male likely have higher susceptibility for the nitro pH. So this is interesting to also see that you see different enzyme, different protein are used or enriched of the patient at the different carcinogen group. So the next question is whether we also see this kind of lay-like patient. And so we compare the proteomic profile again. And again, we see that the lay-like is also layer. They have much higher late-stage patient and higher recurrence. And you could also see that the C2 cluster has a poor survival in overall stage or even more dramatic difference in the stage 1. So that means this kind of lay-like subtype is also present likely in the global manner. And we try to understand why it's happening for the C2 lay-like. What happened? Why is the etiology? What are the major contributions to cause this kind of aggressive phenotype? And again, you could see that it's interesting that we see different distinct groups. The female is mostly for the nitrosamine, and for the male, it's mostly for the nitro pH. And so next, we want to see why it's happening. And this is kind of a summary that even though they are different gender, they have the different genomic KRAS or EGFR mutation. Somehow they share the common aggressive pathway, activating pathway, including Modalities, but they do see difference. For example, for the female, they have the immunoproteasome activation, while the male with the KRAS mutation has the standard proteasome activation. So that just suggests kind of they have a common and unique pathway from the global manners. And this is not just for lung cancer. We also see this kind of heterogeneity in the patient level for breast cancer. I don't have time to go through, but I just want to tell you that we try to focus on the luminal disease, which is the majority of our patient. And we also identify early recurrence of the patient. And we test one inhibitors for the Lumina disease, and you could see that this is much better efficacy compared to the pan-CDK control. And actually, there's a clinical trial failed for this specific inhibitor, and we believe that it's because that clinical trial did not select the right patient, so they average out. Okay, my time's up, and this is the last slide. By this project, that also inspired the development of the single cell, including single cell phosphoproteomic technology for the very first time in the world. And from this, we actually identified that phosphoproteomics could be very useful to identify the driver pathway, including the activating phosphosite. So we have testing on the patient who developed resistance to the OSI. So far we have a few successful cases, and we hope that this kind of technology can be applied for the patient in the near future. And finally, this is my final conclusion. I was very fortunate to have the opportunity to present the plenary talk in the European Lung Cancer meeting. Every doctor talk about biomarker, but it's always at the end. We hope we have the biomarker. So I think proteomics is a very powerful tool, and it can provide the potential biomarker for the different unmet needs. So we hope that we can continue to work for the patient space. And if you need any proteomic technology for your sample, for your project, please let us know. And finally, I would like to thank, this is why I presented is the effort for the big team. So from the left-hand side, they are the brain. We are the workers with the hand, but I think working together is very important. And I always believe that there's no hero, but we do have the good teamwork to generate good results. So I'm hoping that Taiwan can work together for the important questions for the benefit of patients. And thank you very much. And if you have any comments, suggestions, please let us know. Thank you.
Congratulations for the great contribution of Professor Chen. Is there any question from the audience?
For work, congratulations. Regarding the carcinogens, those are from the mutation signature prediction. Is there any lab data to actually treat with this mutagen to the for instance, the initiating cell, type 2 alveolar of lung cell, to see whether it causes mutation or is the exact signature?
Thank you, Raj, for the very important questions. In our first paper, we actually do not do any validation. But in the current paper we submitted, the reviewer asked to do the validation. And so far, we try to use a few cell line to do the carcinogen experiment. But the challenge is there are so many different kind of chemicals even for the nitro pH or the pH. So what we can do is to identify from the literature which carcinogen may be more frequently occurs in our region. So so far we have a few carcinogen like 1-NP, BAP, etc., to do the cell line experiment. But the question is, and challenge is there. Maybe we ask the expert from here. For example, carcinogen like nitrosamine occurs on the female. How can we have a cell line model to mimic the female with the EGFR mutation? That is our challenge. I mean, whether we are able to have a good cell line model or animal model to mimic our patient, I think that is the very difficult part for us.
Yeah. I mean, we can suppose EGFR is the early mutation. Even with so we can start with cells without EGFR mutation, then look at whether this nitrosamine APOBAC is targeting EGFR genes by those chip assays, etc.
Yeah, thank you. That is actually what we are trying to do now. So we have the cell line with the different EGFR or KRAS mutation status. So yeah, so that's ongoing. And actually from proteomic or phosphoproteomic data, we actually see the impact from the carcinogen mutation. So that also provide additional information to that. But the gender, I don't know how to mimic the gender.
Yeah, that's a problem. And you said the nitrosamine is a female imprinted mutagen. It's because it's produced from the cooking or high temperature cooking. But is there any epidemiology to support? Because female cooking so far is not been proved as a risk factor, and so how does this link to young female? It's still a problem which is not solved.
We actually try to ask the expert from NHI because I know NHI has the expert to analyze the carcinogen directly from the blood or urine. So I actually talked to Yu Song Liang just last night. We should add this kind of data for direct analysis of the carcinogen. So if anyone knows the expert, I know Guo Chenqing 老師, right?
Can
so
I add? I think it's some speculation. I think the EGFR is quite clear because the EGFR dimerization, induced the phosphorylation by itself.
I
think EGFR, is dimerization, and you have different mutation may have some effect. And then the other thing also, EGFR, I think in some our studies before shown interact with the I think SH3 domain, SH2 domain, which has interact with all this phospholipid in that way. So that you had mutations, mutation structure study will affect the EGFR activity.
Well, there's a recent work from Jonathan Swanson, and he discovered that the carcinogen PM2.5 may change or affect the oncogenesis degree of the EGFR mutant. So that suggests that carcinogen may change the progression of the lung cancer in vivo. He used animal model, but at the patient level, we also see that somehow they have different susceptibility for different patients. Yeah. But we don't know why nitrosamine is only enriched or important for female. We don't know yet, but we suspect. We also see ApoB enriched in female, but that can be directly linked to hormone. But for nitrosamine, we still feel that this is a puzzle.
It's actually Charles Swanton from UK. He also showed very nicely the air pollutions that have the effect on the carcinogens on the lung cancer and published in Nature. So the PM2.5, and then they are not only for the patient sample, but also they have the mechanism to show that this kind of carcinogen-related carcinogenesis in lung cancer. Maybe because that's the genomic study from the patients and animal studies, then yours is the proteomic. Maybe can look at it and integrate those data to see whether they have the common pathway or effect. Maybe can generate some, and also our member in NHI may have some epidemiology and also this kind of experts to integrate to your study. Thank you.
Yeah, thank you very much for your comment. I don't have time to talk about that part. We actually see the never smoker with a low smoking score has a very similar activated pathway like Charles Swanson mentioned. IL-1, MAP kinase, and also the PD-L1. We do see that in the patient. So that is a further evidence to show what he mentioned is correct. But now with the female, it's mostly enriched in the male.